So audience, today I have to share about my 3D locomotion project for the zebrafish analysis, and here you can see that I will tell you that how this app that will help you for the analysis of the clustering data that you can use. So this is user friendly app. You just have to open the bat file, and here, like you can see, will open, and it will see that whether the dependencies like some of the Python based modules are installed or not. Once, because this is my system and here it's already installed, so you can see the interface is open, and now you have to check about the number of input files, like your Excel data that you already have the tracking data in your folder. So I input the 3, and then you browse, like this is the app folder, and then you check the species. Here is my file, so you can select, and then you will select the next one, and then you will select the next one, and after that you have to select the folder for the output where the all plots or the output file will be stored. So now you can see it will start the processing, and it will take some time, but one thing you keep in mind that while processing you should check that whether your CUDA, like for GPU, is configured or not, otherwise you have to do, and in that point, if it does not work, then it will by default use the CPU. Here it's showing the epochs using Keras. I developed this project, and it will show initial some training, and then you can see how it works. Almost 5 epochs it will show, and later 5 to 10 epochs it will train, and it depends on how much the separation power you entered. Normally 5 to 10, but now you can see it's taking more because I check different separation power, that how you evaluate your clustering methods like k-means, Birch, GMM, or the spectral analysis. So after taking the hierarchy for the initialized training, you can see the clustering result can be displayed. You will select the OK, and now you can see the output in this way. So keep in mind that these output will also be saved in the respective folder that where you selected for the output files. So here is the one figure for the before encoding, and other is after encoding, means one is without deep learning and other is with deep learning. So we used to apply deep learning to enhance the scoring functions for the two statistical methods that commonly used in clustering for the Silhouette score and Davis-Baldwin index. And based on this you can see the separation power, and also you can see the most relevant or most comparative or related values for the locomotion behavior, that which one is more similar, which one is more far apart, something like that, and also the dendrogram to see how the correlation between the index is working. And now I will show you the next part, in which you will see the same process, that you will open the bat file and run the app, and now I will show you the different behavior pattern to see that how the plots are being saved, and then I will run again to show you the different separation powers. So you can see that all of the data, the Excel files and the scoring functions results are already saved in the folder, and also the clustering and the deep learning based analysis for the output all are inclined, and then you can compare also from the Excel files. Here you can see the values. And other thing that is important, that it is like more faster if you analyze data manually. And the other important feature is you do not need to do any kind of like pre-processing for the customization or for the locomotion behavior analysis related to neurodegenerative diseases as well. Zebrafish is well recognized for the neurodegenerative disease model for animal modeling in clinical studies. Now I will run again and select the different data. Okay, same data, and but you can try different data. Now I change the value, you can see from 0.1 to 0.3. I will just show you the difference of the spectral analysis in the separation power. And again you see it's running on GPU because I already configured GPU on my system, but you have to check your system as per the user how it works. Again you have to wait, and you will see the processing later. So this is user friendly app and based on Python, and you can see that the results are based on from sequential analysis, and the output is in like imaging, like you see the different images of the clustering. So the time span and the time steps for the memory already managed, and also depends on the power of the GPU, like this is RTX 4070, some are 4060, so yeah, it varies. And right now they already updated the TensorFlow library, and sometime you will face the problem. I already mentioned the note on GitHub, my source code link, so you can use the PyTorch, and it's already embedded. If you use the PyTorch, then you do not need to install separately TensorFlow library in this extent. And other is like you can see how it's going to analyze fish behavior in different tanks. So this will be really helping for the future studies. This is a concrete data set that mostly we use to analyze, and we check according to different system. And most of the time the number of epochs that you see, it runs using some initial training duration, and also to check the layers of that model, like deep learning models that we used. And it depends on pooling also, and also some other layers, that how the machine learning model is fetching the data. Now you can see last time, if you remember, you can see my tutorial. If you go back, you will see like it was ended on 19 epochs, but here you can see the more epochs. Why? Because I changed the value from 0.1 to 0.3 for the separation power. It will show you the more dense clusters for the data sets. Most probably it depends on type of data sets and also the species. For example, if you want to analyze other zebrafish species or strain, or the other zebrafish strain and also other species, for example in different aquatic animals, but this data set is trained based on zebrafish data. And now you will see the output. Potentially it varies. Sometimes feature selections affect also, sometimes the fine-tuning luck. Yeah, now you can see the cluster after encoding, it's more dense. So this is what I want to show, the consistency between the clustering models, that how the results before and after deep learning, it varies. So most of the time you can see the results that are automatically saved, but one thing, you should delete the previous files that if you want to change the separation power in the next round, otherwise it will be replaced. But sometimes the app will have some, like while replacing the files it will show error, so it's better you should delete the previous results, and then when you run the app again, it will save the results again with empty folder.